I am here to tell you about the experience of being an early adopter of a technology. As one who started a company like Luminex, I was going around the world, especially to One Kendall Square, trying to sell Luminex instruments in 2000. And trying to get people to adopt the new technology is really hard. It's really hard. If you've got the right technology, it happens. And what you need to find are the early adopters, the people that can not only fall in love with the technology, but quickly get it to market, commercialize it, and show people that it's a commercially viable technology. And that's exactly what we've done. I guess we've known Kevin and a lot of his crew for many years, going back to the Perk and Elmer days and trying to sell Luminex instruments there as well. So it's been a good relationship, and so we were, being propeller heads, we're always interested in new, especially immunoassay technology. And when we first looked at Quanterix, it was just fabulous, and it's gotten nothing but better through the years, and I know it's going to continue to become a very important immunoassay technology in the world of diagnostics. So a rather complicated title, but it really is, as you'll see, it's going to complete our immunoassay offering and our offerings to pharma. We have two missions at Myriad RBM, and most of the talk's going to be about the first one, and I'll save the second one for the end. But what's our first mission? Well, it's to help pharmaceutical companies, biotechs, develop better drugs, more efficient drugs, drugs with lower toxicity and more efficacy. So just to tell you a little bit about the company, we're really founded as a division of Luminex in 1998. In 1998, we realized that the Luminex technology was actually going to work, and we started thinking about the killer applications. And if you take yourself back to those days, everything then was genotype, genotype, genotype. We're going to sequence Craig Venter's DNA and answer all the medical mysteries. Well, obviously that didn't quite work. We never believed that. We've always been phenotypic people, whether it's on the protein side or the nucleic acid side with transcriptomics. We believe that the best application for multiplexing was going to be in the area of immunoassays. So we spun out. After 4 years, I was chief scientific officer of Luminex, one of the founders, spent $25 million of their money, well, it was our money at the time, we had raised in an IPO, to develop the platform I'm going to show you. And in 2001, we showed it to the board of directors, and they said, "What are you going to do with that?" And we said, "We're going to change the world." And they said, "We don't change, that's not our business. We don't change worlds, we sell widgets, we sell instruments." So we said, "Well, can we just spin the thing out? Can we take it out? You know, get it off your hands, we'll get out of your hair." And that's exactly what we did in 2002. 16 employees, went 2 exits down the freeway in the Texas model in Austin, and built a much nicer lab than Luminex has, even to this day. We built this platform, I'll show you in just a minute, and it served us and it served our customers, our biotech and pharma customers very well over the last 14 years. And it's basically industrializing Luminex. No one else has done this. As you'll see in our production lab, we have 42 Luminex instruments running 16 hours a day, sometimes 7 days a week. And they're supported by 8 Tecan Evo robots. So all the liquid handling is done by robotic transfers. This testing business, this is our first mission, to help pharma. And in return for that, pharma writes nice checks that come to our bank account in Austin, Texas. And it's that revenue that we then generate our own diagnostic projects with. And that's the second mission of RBM, and I'll tell you about that just briefly at the end. We have our own internal diagnostic initiative group. I got a little ahead of myself there, that really they serve as a customer for the same platform I'm going to show you. Austin, Texas, 100 people. We were acquired by Myriad Genetics about 5 years ago, and it's been a wonderful marriage for us. You know, resistance is futile. You will be assimilated, but it's really, they're culturally very similar to ourselves, and so we've integrated into their programs very, very well. It's a really good group of smart folks. I don't think I have to explain XMAP technology to anyone here. You know, 100 different colored microspheres, retina flow type instrument with 2 different dyes systems. It's probably, at least in the immunoassay world, with the exception of the ELISA plate, the most heavily adopted and rapidly adopted technology on Earth. So there's almost 20,000 of these instruments placed around the world now. And I show that slide primarily to remind you that we've been building immunoassays on microspheres since about 1994. So we've got a lot of experience putting antibodies onto carboxylated beads. It just turns out the bead for Simoa is much smaller. It's about 1 micron instead of 5. So this is what our production lab in Austin looks like. You can see the banks of Luminex instruments supported by the TCAN Evo there in the middle. And that's where our assay precision comes from. I mean, Luminex is automatically a very precise technology, but you still got to have precise liquid handling up front. We've developed over the years over 300 assays, and these assays have all been developed under contract with mostly pharmaceutical companies, some of the smaller biotechs. We have a controlled calibration strategy on every plate. It's very important to us that the quality of this data is top-notch. Now over 80% of our testing is on phase 2 and phase 3 clinical trial samples, which are incredibly valuable. So you don't want to mess up with that data. I can't emphasize this enough. A lot of people get into the immunoassay business and they forget about one of the most important things in serum and plasma and urine and cerebrospinal fluid, and those are matrix effects. Those things that are known like rheumatoid factor and human anti-mouse antibodies, some of the ones we know, but there's then many we don't understand as well. And if anyone out there with a testing lab tells you they can block all matrix effects, they're lying. No one can. But we feel like with all these years of experience, we have enough cocktails of blockers. We can block about 95, 98% of them. That's why we were really enthused when Simoa came along, because now we could apply all of these years of expertise in immunoassay development to this wonderful platform. Why is multiplexing important? Small sample volume. Of course, it saves money. It's, the most expensive thing in a lab is labor. So if you can run 10 assays in the same well you used to run 1, suddenly you're much more efficient. But it also saves sample. And think about how precious these clinical trial samples are. 100 microliters here, 200 microliters there. It's a point in time they'll never get back. And they might have spent $100 million on the trial or more. So it's a very valuable sample. So the more information, quantitative information they can get from that sample, the better. And it is an FDA-cleared instrument now after all these years. Sorry for the infomercial. I just can't help it. My old sales side always goes that way. But we're now over 700 publications in many different areas. You won't be able to see this, but this is where these publications are found in different disease therapeutic areas. And by far the largest is inflammation and autoimmunity. Cardiovascular is big in here, neurology is big in here, a number of different areas. You can see when you're looking at 300 different assays that are measuring the results of dozens of pathways, it's applicable to many different disease therapeutic areas. Again, we've been doing it for 14 years, a lot of experience. We average about 90,000 samples. I think we did 91,000 samples last year. 90% of those are for this fee-for-service model to help pharma develop their drugs. But then about 10% of them are for our own diagnostic initiatives, as I'll show you. Again, Myriad Genetics, the acquisition has been wonderful. This is their mission statement. They now have done over 2 million BRCA analyses over the years, and of course, I think the Angelina Jolie situation was discussed this morning, and nothing could be more important, we believe, from a blood-based biomarker point of view than to be able to develop a test that a woman who's BRCA1 positive, BRCA2 positive, familial history, she has a 1 in 6 chance of developing breast cancer. I mean, it could be 1 in 3, depending on which study you read. Now, what do you do about that? Well, you know what Angelina did, pretty drastic. But if one had an early stage blood test to detect that early cancer, I think that would be a very good, very important development. They're the largest molecular diagnostic lab now. They're the global leader in hereditary cancer with, of course, all the years with BRCA. They really have been a pioneer, of course, going to the Supreme Court 3 years ago, spending millions of dollars on court fees and losing, so that all the other DNA patents in the world all went out the window. But at least we tried. We went down fighting. A trailblazer in autoimmune disease. We were the first acquisition. The second acquisition was Crescendo Biosciences from South San Francisco. They have a blood-based test that's called Vector DA for disease activity, and it tells you the disease activity of rheumatoid arthritis. It's 12 proteins in the blood, actually measured with mesoscale technology, which is also a very good multiplexing technology. But it measures those 12, applies an algorithm, and then tells a physician and the patient, how active is your disease? Should we treat you very aggressively, or should we back off? About 35% of rheumatologists in the US are now regularly using vector DA. It just validates the whole idea that came up several times this morning. It's not a single biomarker when you're looking at protein in the blood. It's a pattern of biomarkers. And they made quite a bit of money last year. In fact, we just used some of those revenues to acquire the third acquisition of the company called Assurix Health from Mason, Ohio. You may know them. They're a genotyping company that actually uses the genotype of an individual to determine what's the best antidepressant medication to put someone with major depressive disorder on. So what is our approach with RBM? It's the same whether we're using a pharmaceutical customer or ourselves. We start by casting a wide net as possible. We go in thinking we don't really know what we don't know. So we cast as wide a net as possible. We identify a key pattern, and then we do what we call shrink wrap. We're going to shrink wrap. Let's say we test 350 assays, and we find that only 10 really can distinguish between drug-treated and not treated, or ill and not ill, and we'll shrink those 10 into its own little multiplex. So the advantage here is it's one validated, and when I say validated, I'm very serious about that. These are very heavily validated assays. This is just a fun graphic that we've used for years. We're comparing immunoassays the way we used to do it, which is by ELISA. You know, it's one at a time. It's kind of a rod and reel method. But with Myriad RBM, we just catch the whole school, flop them up on the deck, and see which ones are important. Now, it's important to look at this graphic. If this were 15 feet deep, there's, it's kind of analogous to the 15 logs of protein concentration in the proteome, right? With ELISA, mesoscale, Luminex, every other immunoassay technology I can think of, we can see down in this pool down about 12 feet, about 12 logs. But once we're 3 feet from the bottom, it becomes very, very murky. And what you're hearing today, and you'll hear a lot in the future, is there's some very important targets there. The fact that we can now measure some of these inflammatory and anti-inflammatory molecules in the blood in normal individuals where we couldn't before is a real breakthrough. So we were an early adopter. I think we purchased our first instrument about 1.5 years ago. And we had a target to launch our first assays in January, and our group did a wonderful job applying all that know-how from years with Luminex beads onto these wonderful little Quanterix microspheres. And we now have 3 instruments. As you'll see, we actually built a new lab just for the Quanterix instruments, and we built it about 5 times bigger than we needed because I think we're going to buy that many instruments in the future. We use one for assay development. So just to be clear, we develop these assays ourselves and validate them to the standards I'll show you in just a moment. We currently have 11 assays with about 4 more. These are all singleplex now with about 4 more, and that's actually kind of an old slide. I think it's up to about 6 or 7 assays. Again, everything is contracted by pharma. They want these molecules for very specific reasons, usually for pharmacodynamic analysis. And I'll tell you about multiplexing at the very end. Immunomodulators are probably the biggest area right now. I think it's going to continue to spread. And when I say immunomodulator, that's immunotherapy like checkpoint inhibitors in pharma for oncology, but also in inflammatory diseases like RA, ankylosing spondylitis, psoriatic arthritis in the autoimmune space. So what do we do when we build a Simoa assay? It's really very simple. Most of these assays we've already developed on Luminex, so we simply port the antibody pair over. Usually that works. Sometimes we have to go out and find a different antibody. We often have to use a recombinant calibrator, and that's something that a lot of people don't think about. In most of these assays, we're calibrating, and in some cases even controlling the assay with a molecule that is not exactly like what's in the blood. And it causes a lot of problems. So you have to be very careful about your calibrator screen. This, again, I can't emphasize enough. There's so many matrix effects that one sees in serum and plasma especially. We go through an enormous screen with about 12 different cocktails of different blockers to try to minimize the number of matrix effects. Once we feel like we have a prototype assay, and that's what I'll call it here, a prototype, We're then going to do the following. We're going to look at normal serum. These are people that are fairly young people typically that don't have any chronic diseases. We're going to look at plasma because there's often, as you'll see, differences. We're going to look at disease-based sera, and we're going to look at heterophile sera. So those are sera that we already know have these matrix effects in them. And then finally, the third leg of our immuno monitoring platform for pharma is something called True Culture, which I won't have time to go into today. But it's a point of care, whole blood culture and collection device where one can monitor what somebody's immune system's doing in real time. It's a lot of information on our website. If you're interested, take a look. So we'll also use True Culture supernatants. These are actually little supernatants out of this tube because they have very high concentrations of native cytokines. So we want to make sure, can we measure, even though we're calibrating with a recombinant antigen, can we measure the normal molecule at very high concentrations? Okay. We always correlate back to the Luminex assay where we can, and then proficiency tests with an ELISA. And this is because it's a requirement, as you'll see, of CLIA, being a CLIA certified lab. Antibody competition, this is to make sure that the antibody is actually binding what you say it's binding. And it's another step that some people like to skip. And then finally, once all that's done, we're ready to do what we call our full validation package for a kit. And this kit may work for 2 years worth of samples, depending on how many samples come through the lab. And this is our new lab with our 3 Simoas, one you can see there on the back. But the first thing we're going to do is determine the sensitivity, doing LDD and LLOQ, least detectable dose and lower limit of quantitation, as you'll see. We'll do the normal and dynamic ranges. These assays tend to have enormous dynamic ranges. They really do. We'll do imprecision, spiked recovery, and linearity. Now, the imprecision, of course, kind of speaks for itself. You want to make sure that you're getting somewhere above or below 20% coefficients of variation with these assays. The spiked recovery and the linearity both have to do with matrix effects. Because if you're looking at linearity, as you dilute a sample down, your results should go down as well, right? Or if you add a set amount of material in, of antigen in, you want to be able to collect, basically 100% of it. Again, a problem is that we're using recombinant antigens, which can certainly complicate spiked recovery. Correlation is always done back to ELISA. That's a CLIA standard. Cross-reactivity is something we look at very carefully, especially when you start to try to multiplex. And then the matrix effects, as I mentioned, and stability studies. So all of this is really a requirement for one to have a CLIA-certified immunoassay lab that services phase 2 and phase 3 clinical trials. If you don't go to this level of validation, then you're on your first audit from Pfizer, Merck, or AstraZeneca, you're going to really get dinged. Okay, this is what our menu looks like now, and I hope you can see these in the back. Simoa LLOQ, and this is Luminex. And you can see this is the fold increase in sensitivity, improvement in sensitivity. Some of these, like IL-17A and IL-12P40, these probably could be better, but what we're trying to do is make it so that when we look at a population of normal individuals, we can measure them all, or nearly all, toward the bottom of that curve, giving us a huge dynamic range. I'll show you an example in a minute. But the one that I would point up to here is interferon gamma. This has turned out to be a wonderful assay. As you can see, 263-fold, and we measure virtually everyone who's normal. And this is an assay that's in just huge demand, right? And we can't make it fast enough. If you took the average there, it's about 300-fold. So the technology is as advertised. It does it, and it does it reproducibly day in and day out. Just a couple of curves. I don't want to bore you with a lot of curves here, but this is the BLC or CXCL13 marker. And again, we're looking here at the lower limit of quantitation, which is 64 femtograms per mil. I didn't know if I'd ever be able to stand up here and talk about LLOQs in femtograms per mil. And upper 130 picograms. So look at that dynamic range. Here's where the normal samples are falling, right? And that's kind of where we want them to be. We don't want them to be too far down the curve. We want them to be somewhere in this range so we get a lot of range so we can look for pharmacodynamic results. This is typical, same marker of an imprecision run. We'll run it 5 times over 5 days with a series of different operators to see what the cumulative inter-run CV is, and this is pretty typical. Again, that's a tri-level control. So again, this is a serum-like or plasma-like fluid that we then spike in 3 different levels of the control of the antigen. Again, often a recombinant antigen. Now, this is something that people don't think about sometimes when they come to us all the time. We've been working with these groups. We're supporting hundreds and hundreds of clinical trials right now. The first question out of their mouth is, should we use serum or plasma? You know, it's the #1 question that our customers have. And it's something you need to address very early and very carefully. And let me show you some examples. Interleukin-6, a very common inflammatory marker. Here, this is plasma, box and whiskers, and this is serum, box and whiskers. Very similar, not a big difference in IL-6. Interferon gamma, you start to see a little bit of difference here. You'll see where plasma actually has a little bit better representation of interferon gamma. Again, this is minor, but you can see that if you ran plasma that's partially clotted, you're going to have a very imprecise study. And it can get worse. Here's interleukin-10, a classic anti-inflammatory marker. Look at the difference. Serum, plasma, and then finally, this is the worst offender of the cytokines we know of, IL-1 beta. Look at that. It just disappears. MMP2 and MMP3 are the same way, although they're yin and yang. One's in serum and one's in plasma. So you really have to ask these questions up front. As I mentioned, the first thing we'll do during validation 2 is look at disease state samples. And unfortunately, I'd love to give you some anecdotal stories, but everything we do for our pharma customers is under strict CDA, as you can imagine, supporting these huge clinical trials. But we do on occasion do some of our own internal studies. For example, in rheumatoid arthritis. Here's rheumatoid arthritis for interleukin, again, interleukin-1 beta. And you can see, obviously this is plasma. You can see that there's a bit of difference in the average. This is actually the median of the box plot, which might be a little more significant than the picograms per mil, but they're basically doubling in the RA. Again, looking at 44 femtograms versus 80 femtograms. So is that significant? I don't know. It would have to be a pretty large study to determine that. TNF-alpha, let's see, TNF-alpha, a little bit bigger difference. Now we're up in the picograms here. 1 picogram for the normal, but look, almost 4 picograms in rheumatoid arthritis, and certainly some outliers, which are probably skewing this median up some more. And then interferon gamma. As it turns out, interferon gamma is not a real big marker in rheumatoid arthritis, at least in our hands. Okay, finally, before I get to the last mission, we're just starting the multiplex. We've launched all these assays as singleplex assays, and they each use somewhere around 50 microliters of sample. We're working on that. We're trying to get that volume down because that's key for pharmaceutical customers. But the biggest key is multiplexing. And our first, again, contracted by a pharma, TNF-alpha, IL-6, and IL-17A. And you can see basically the results. What we ask are that the results with each one of these are essentially the same as when they're running singleplex. That's always been something that demand is what we've put for years on all of our Luminex-based assays as well. Okay, so I hope that gives you a flavor of what we do and what we have done to fairly rapidly port some of these very important cytokines, chemokine assays over to the Quanterix Simoa platform. It's an amazing platform, and I hope you have an opportunity to generate data using this in the near future. Now, the final mission, from the very beginning of RBM in 2002, actually in '98 when we dreamed this whole thing up, the idea was to use the fee-for-service business to fund our own diagnostic initiatives. And for many years, we've been interested, I guess we should now be in the neurology track, in psychiatric diseases, schizophrenia, bipolar disorder, and of course, major depressive disorder. And in a study actually started, I guess, about 6 years ago, it was before we were acquired with the Mayo Clinic and the University of Minnesota, we were able to tease out a signal using a combination of proteins, which addresses what we believe is the most difficult diagnostic problem a psychiatrist has. And that's when a person presents as a depressed individual, there's a 1 in 7 or 1 in 10 chance that they're not major depressive disorder, they have bipolar depression. And the problem there is if you present that or treat that person, prescribe that person a very strong antidepressant, it can kick them into the manic stage of bipolar and they go home and they drown their kids. This is a very serious diagnostic paradigm. It's something that over the years we have learned that this is what a psychiatrist is the most scared of. A principal care physician is enormously scared of it because he or she doesn't feel like they know enough. And so that's why major depressive and bipolar depression is often treated with sort of kit gloves to begin with. We believe we have a pattern that we can differentiate these two diseases with an area under the rock curve about 0.9. We're in a 5-site prospective validation trial right now being funded by Myriad with about 300 patients. We hope to have all of them enrolled by the end of this year. And this may be the first contribution of RBM to Myriad proper. And it's really the reason Myriad acquired us in the first place, is they would like to not just be a one-trick pony with BRAC analysis, but to branch out into some of these other markets, and certainly neurology and neuropsychiatric disease. There's not a bigger market than that. So that's our second mission. And I hope that gives you an idea of where we are with this technology, how much we appreciate it, how much we appreciate these guys. We can't tell you how many times, middle of the night, we've called and said, I can't get the instrument to work. I mean, they've been amazingly responsive over the last 18 months since we got our first instrument, so we really do appreciate that. Okay, any questions?
I have a couple questions. One's related to your clinical trials business and one to your diagnostic business.
Okay.
Clinical trials business, as you're growing this business and you're doing, supporting clinical trials, presumably you look at patient stratification, disease response, and so forth. Is there a path for some of those to migrate over to a true companion diagnostic? And if so, how are you thinking about the IVD and access? So that's the diagnostic question.
Okay. Okay.
The diagnostic one is just expanding upon your next areas of interest. You mentioned neurology, and obviously you have a big inflammation, and there's lots of expansion.
Yeah.
What other, say, non-oncology, so everybody is going in that area, areas are you looking at? So
For our own diagnostics, let me take the second one first. For our own diagnostics, because of the acquisition of Crescendo, we have a very active program in RA, psoriatic arthritis, psoriasis, and ankylosing spondylitis, and we're trying to get into lupus because we already have a sales force. Right? So we've got a business unit there. That's one of the biggest. We've always been very involved. I don't know if anyone's seen the Origin study from Sanofi. That was published about 2 years ago, and we did all the testing for that. That was 8,800 type 2 diabetics. A lot of them have developed diabetic nephropathy. So we have great interest in nephropathy and retinopathy. That being said, those are the main ones. Now that we have acquired Assurix Health, we're going to be back into depression in a big way. As far as your first question goes, how do we, and could you rephrase that?
yeah, I'm just curious if for some of those relationships with pharma, are any of those going to turn into a companion diagnostic type opportunity? Because there's a fine line, right?
It is.
If you stratify the population too much, the FDA might force them to put it in a label, in which case then they need a platform. And since you're really LDT, not IVD,
it's a great question. I apologize. I can't multitask very well, as my wife says. Yeah, I mean, look at Keytruda and Opdivo. Yeah. What a great example of a company pigeonholing their market. And yes, we're in development with a number of pharma where we will collaborate with them and we'll do either reduced price testing or free testing for free with the idea that if a companion diagnostic opportunity happens, we'll have the first shot at developing it. Now, your question gets much more deep when it gets to IVD versus LDT. We always at Myriad are going to take the LDT route. It's the quickest to market. It's the best value chain. But you're going to want worldwide distribution. So the LDT will win out, but we're just waiting to see the first really big one. The other problem is who pays for it.
Exactly.
Yeah. Yeah.
So to follow up a little bit on that, as you develop the product and you do go to the companion test, in terms of IP protection and ownership, especially with your pharma partner, how do you strategize those discussions, and how important is it to you to be able to capture the asset, the content of the asset?
Yeah, that's really, I guess, two questions. On the IP, we typically tell the pharma anything that has to do with the drug, obviously, the drug or any way to stratify your patients, you own that. You will give us then license, if we can develop a companion diagnostic, you'll give us the license to develop that, or at least the first shot, first right of refusal to develop that. And in lieu of that, we will actually share, the IP for the companion is typically theirs, but then we have a commercialization right. Does that make sense? Because pharma does not want to give up any IP, I guarantee you that. Yeah. Did I answer the question? Okay. Yeah.
Ralph, you talked about the similarity of Luminex and Simoa and how everything kind of falls. Have you seen any differences from measuring the high concentrations to kind of below 12 feet, or does everything kind of scale? Are there new challenges?
Yeah, no, good question. Good question. This goes to the point of view that now when a customer orders a map, one of our most common multi-analyte profiles is called inflammation map. It has 46 assays. Well, it has some of those 12 as well. So what we decided as a company, when we report back that 46, if you've done any Simoas, we're going to give you the Simoa data. And the reason is most of those Luminex assays, we're way down on the curve, so our imprecision is much higher. Obviously, it's not as good an assay on a less sensitive box. So we're going to give you the Simoa assays. If people ask for both, we will, but we warn them you're going to see differences at the lower end. Now, if you're on the upper end, middle of the curve, and further up, they correlate very well. It's just on the bottom of the Luminex curve. Does that make sense?
I guess my question is more the fear of the unknown. Is it really different down there?
Oh.
The measurements and things you have, the challenges you have, or is it just the same as higher? Is there anything new?
Yeah, I wish I could tell you some of the things, but I can't. There's also another interesting phenomenon we've seen, and that is a pharma coming to us and saying, I know your Luminex assay is very good where we're measuring it with Luminex, but we know our drug makes it fall into the murky 3-foot depth. So we want you to develop a Simoa assay to measure after drug treatment. And in fact, we'll use the Simoa assay both before and after. And there's more of those than I think we thought. You think of disease making things go up, but that's not always the case. So yeah, I think it's a whole new, the horizon is so blue here of the things that we're going to find. It's just like the early days of radioimmunoassay. People said, we don't need to go way down there. Why do you want to measure, picograms per mil? And look what we found. So there'll be a lot of good fish in the bottom of that lake. Okay?
Okay, great.
Thank you.